Okay, guys, I thought we could try chatting today via, well, I started a chat, I think. So we could try that method today. See how that works. Let's see. Okay, Crest, I think this is who is that? Can't remember your name offhand, but anyway, yes, we're gonna chat this way today. Okay, so I see your responses, and we could use this method. Okay, so people are still signing in. Let me get those. Are you guys seeing the chat? Can we use the chat today? I see Abraham, I know you've seen it, and Crest, I know you've seen that. The others. Can you guys let me know if you're seeing the chat? Okay, I see that. Okay, I see those. Most of you have seen it. Okay, this is good. I will give it about 2 more minutes for the rest of us to sign in. Charlotte, you posted. What are you saying? Let's see. Okay, Charlotte, you're here. Got you. Okay, Ezra, I got you. Okay, so it's 10:32. I think we need to get started. Hannah, okay, great. I think we're good. My phone is just, let me turn that off. So today, let's see. I want to show you a slide. Let me see if you see it, okay? Let me know. Oh, I missed the slide here. Okay, can you see this slide? Okay, you can see the chat. Can you see the slide? I just placed the slide up. Okay, great. So we're looking at Meto and Tracker. Okay. These are two primates that were created from nuclear transfer, oocyte nuclear transfer. So what is oocyte nuclear transfer? Last week, we talked about mutations in the mitochondria, and we said that mitochondrial mutations are inherited via maternal inheritance. So mutations in the mitochondria are passed on via the mother and not the father, because the egg, the gamete, female gametes are larger, so they have more mitochondria, so they pass on. They have a higher chance of passing on mitochondrial diseases. So if there's an individual with a mitochondrial mutation, to bypass that, what you could do is what is known as nuclear transfer, oocyte nuclear transfer. And let's take a look at that. Here we have a mutant mitochondria. So this is a mother containing mutations in the mitochondria. And to bypass that, what you could do is an oocyte nuclear transfer. So here you remove the nucleus of the mutant mother. Here you remove the nucleus of the mutant mother. The mother's nucleus is removed. And here's a wild type individual, so normal cells are here. Well, normal cell, but normal mitochondrial DNA is present here. So you have normal mitochondrial DNA. And here you remove the donor's nucleus. So you remove the donor's nucleus. Then you take the original mother's nucleus and put it into the donor's egg. Okay? This is the oocyte. Put it into the donor's egg. And that is how nuclear transplantation takes place. And this allows you to sidestep mitochondrial diseases. So if you recall the mitochondrial diseases we talked about last time, this is how you would sidestep that. LON, LOHN, we talked about that. MERF, we talked about MERF. These are mitochondrial diseases that occur in humans. So to sidestep that, you could perform these nuclear transplantation. And that's how these two guys were produced. Okay? All right. Okay, you're saying the slides are frozen. Okay. Well, these slides, let me let you know, this slide was from last week, so you should have these anyway. So I'm going to transition now onto chapter 15, which is the regulation in prokaryotes. Heaven, yes. Use the slides on Moodle. Use the slides on Moodle. This is from last week, so don't get too confused with this. I'm sorry. This is from last week. You already have those slides. I was just finishing up the chapter. Now we're going to transition to chapter 15, which is gene regulation in prokaryotes. Right? So let's move there. For those of you who are having problems getting on Facebook, I have a poll everywhere link there, so you could communicate with me that way also. So look for that on Moodle. If you're not on Facebook, you could use that method. Okay. All right, let me get today's slide out. Okay, how about this? Can you see this slide? It's regulation of expression. It's entitled Regulation of Expression Can Occur at Many Steps. Let me know if you're seeing the slide. And I just need one person to respond. Okay, great. So we're talking about regulation of transcription, no, sorry, regulation of gene expression in prokaryotes. And we learned that there are two phases of gene expression. There's transcription and there's translation. And within those two stages of gene expression, that's the transcription and translation, you have initiation, elongation, and termination. And so regulation of gene expression can occur at any of these steps. We know that the enzyme responsible for transcription is RNA polymerase. And in the past, we looked at how RNA polymerase initiates transcription. We looked at the factors necessary for transcription, the factors necessary for initiation, elongation, and termination of transcription, and how these the role each of these factors play in transcription. So bearing that in mind, know that gene expression is regulated at the level of transcription. So it shows you here transcriptional control occurs in gene expression, and it can occur at several steps. It can occur at the step of binding of the RNA polymerase to the promoter. Can anyone tell me what is the at what stage is? Oh, let me ask the question. What are the consensus sequences to which the consensus sequences RNA polymerase binds to in the promoter? Does anyone know that? At least one. Name one of the consensus sequences found in the promoter, or the location, I should say, of one of the consensus sequences found in the promoter to which RNA polymerase binds. Anyone? Yes, the Tata box. Very good, Heaven and Talissa. Tata box is one of them. And the Tata box is in the -10 region, and then we have another in the -35 region, another consensus sequence. Okay, very good. So we know at that step, transcription could be regulated at the binding of the RNA polymerase to the promoter. For example, you could block that sequence on the gene, and that will prevent the RNA polymerase from binding. So that would be one way of regulating gene expression. So this is the most critical step in prokaryotic genes, that binding of the RNA polymerase to the Tata box. Also, the shift from initiation to elongation is another area where expression could be regulated. And once the messenger RNA is made, termination has to occur. And we talked about those factors involved in termination in the past. We said there's rho-dependent termination and rho-independent termination. Rho is a protein that helps to facilitate termination. So that rho could also be involved in regulating gene expression, because that is where termination occurs. Well, it's involved in terminating transcription. So without termination, which is the release of the mRNA at the termination site or in association with rho, you could affect the length, for example, of the messenger RNA that is produced, and the content of the messenger RNA that is produced. Okay, regulation could also occur post-transcriptionally. For example, the stability of the messenger RNA, how long a messenger RNA stays in the cell. So the longer the RNA stays in the cell, the more protein that you could get from that RNA. So example, if a messenger RNA is made and it's readily degraded, then the amount of protein that is produced would be low, I should say, compared to one that allows to an mRNA that stays around in the cell. So the longer the messenger RNA stays in the cell, the more protein that is produced. So the stability of the messenger RNA is key in regulating transcription. The efficiency of translation initiation is also important, and the stability of the protein that is produced. If the protein that is produced is readily degraded, then it doesn't do much for the cell. So it's important that the protein that is made after translation stays around for a while. Okay, so these are just some of the steps at which regulation can occur. Let me change this a bit here. Okay, so in a cell, we know that in a cell metabolism occurs, and regulation of metabolism really occurs at the level of transcription or even translation. And metabolism is divided, I should say, into two phases. You have what is known as catabolism and anabolism. And these two pathways require different types of gene regulation. So catabolic pathway is where molecules are broken down. So for example, the ability to utilize a molecule, the ability to utilize glucose, for example, the ability of a cell to utilize glucose, the ability of a cell to utilize lactose, or any sugar you can think of, requires catabolism, breaking down of that sugar or molecule into components that are useful by the cell. So catabolism requires an inducible type of regulation. Inducible means that the genes that are necessary for breaking down these molecules are turned on only when these molecules are present. It will be very inefficient for a cell to have these molecules turned on to full expression if they're not needed. So only when a cell, for example, bacteria, is in the presence of a catabolite, for example, if in the presence of glucose or in the presence of lactose, only then will the genes necessary for breaking down the lactose or the galactose would be turned on to full expression. Okay, so catabolic pathways involve the breaking down of molecules and require an inducible type of regulation. So regulation is only induced in the presence of these catabolites. Unlike anabolic pathways, which involves the build-up, the synthesis of molecules from a simple constituent to larger ones, this type of regulation requires what is known as repressible regulation. So example, your amino acids are examples of pathways or substances or molecules, I should say, that require anabolic pathways. So cells when make these amino acids when necessary, and in order to make these amino acids, anabolic pathways are used, and anabolic pathways require repressible regulation. So in repressible regulation, the pathway is turned off. So the pathway for making amino acids are turned off unless the cell needs it. Okay, the pathway for making amino acids are turned off unless the cell needs it. If the cell needs it, then the pathway is turned on. So cells, bacteria, we're looking at bacteria, prokaryotes are very inefficient. They're very efficient in how they regulate genes. Genes are turned on or turned off only when necessary. Okay, so we will focus first on a catabolic pathway. So we will spend this lecture, we will look at a catabolic regulation, or we'll look at regulation of a catabolic pathway using the lac operon as our example. And the lac operon uses an inducible type of regulation to regulate the components of that operon. So let's see. So lactose utilization. So lactose is a sugar that is used by prokaryotes. Specifically, in this example, we're looking at E. coli. In order to, for the cell, in order for bacteria to be able to utilize lactose, the cell requires an enzyme known as permease. Permease is necessary, and also beta-galactosidase. So permease is involved in transporting lactose, or forms a passageway, I should say, via which lactose can cross the membrane. Okay, so a cell, imagine a bacterial cell in media that contains lactose. In order to be able to use lactose, lactose has to cross the membrane and get inside the cell, and it passes through the membrane via permease. So permease is necessary for the utilization of lactose in E. coli. Once the lactose gets inside the cell, it has to be broken down to smaller components. In this case, it's broken down to galactose and glucose. And in order to do that, the enzyme beta-galactosidase is required. So beta-galactosidase is breaks down lactose into smaller components. So this is an example of catabolism, the breaking down of a larger molecule into a smaller molecule. Okay. In the absence of permease and galactosidase, beta-galactosidase, the enzymes, in the absence of lactose, if lactose is not present, these two enzymes, that is the permease and the beta-galactosidase, they are present, they are on, they will be there in the cell, they're present in the cell, but they're present at very, very low levels. They're present at low levels. So only when lactose is present would these enzymes, or the genes for these enzymes, would produce higher levels of permease and beta-galactosidase. So lactose acts as an inducer. It acts as an inducer of the genes for permease and beta-galactosidase. Okay, if you recall, we said that, let's go back to the catabolic pathway here. Catabolic pathways require inducible regulation. The pathway is turned on only when molecules to be broken down are present. So in this case, only when lactose is present would the components of this pathway be turned on. Okay, and we're seeing that here. Okay, so only when lactose is present would permease and beta-galactosidase be turned on to high levels. So lactose acts as an inducer of the lactose utilization pathway. Okay, let's move on. Okay, So what we know of lactose utilization was done by studies conducted by Jacob and Monod, and they study what is known as the lactose operon. That's abbreviated LAC operon. An operon is a cluster of genes that function together as a unit. So in the bacterial chromosome, in E. coli, but not just E. coli, but all bacterial chromosomes, you will find that genes occur in clusters. And these clusters of genes are expressed and regulated as one unit. This one unit is referred to as an operon. So the LAC operon or the lactose operon is used to it's made up, I should say, of a cluster of genes and regulatory units that are necessary for the utilization of lactose. So if bacteria is placed in media that contains lactose, then this operon will then express the genes that are necessary and regulate those genes that are necessary for the utilization of lactose. So let's look at the components of this operon. First of all, the operon contains 3 what are called structural genes. And structural genes are genes that make protein. That's the basic definition of a structural gene. A structural gene is a gene that expresses a protein, that has the information for making protein. Not all genes make protein, because we know some genes make ribosomal RNA, and ribosomal RNA never becomes protein. We know there are genes that make tRNA, and tRNA never becomes protein. So structural genes are genes that encode information, I should say, for making protein. So the lactose operon contains 3 structural genes. These are they're called LAC-Z, LAC-Y, and LAC-A. LAC-Z, LAC-Y, and LAC-A. LAC-Z stands for beta-galactosidase. We saw that. LAC-Y stands for permease. That's LAC-Y. And LAC-A stands for transacetylase. And all 3 are structural genes found on the LAC operon. And so this is the LAC operon showing you the 3 structural genes. In addition to the structural genes, the operon also contains an operator site, which is adjacent to the LAC-Z gene. So there's operator site and a promoter site. Okay, so promoter, operator, 3 structural genes. The operator site is where RNA. The promoter site is where RNA polymerase binds. So we know that RNA polymerase binds to the promoter. The operator helps to control initiation of transcription, and we'll see how it does that. Okay, so we have promoter, operator, and structural genes. The role, I should say, of each of these components, of all the components that I mentioned of the LAC operon, the role of each of these components were determined by creating mutations. So in studying this operon, Jacob and Monod, what they did was to induce mutations in each of these sites, induce mutations in the LAC-A gene, the LAC-Y gene, the LAC-Z gene, the operator site, the promoter site. And based on how lactose was utilized in the presence of these mutants, it was determined the function of the wild type gene. So mutations helped in determining the functions of these wild type genes, of the components, I should say, not the genes, but of the components of the LAC operon. Okay, showing in this figure, you'll see that in addition to the structural genes and the promoter site and the operator site, we have here a repressor protein. That repressor protein is encoded by a gene known as the LAC-I gene. And the LAC-I gene encodes a repressor protein here. And that repressor affects how the genes, the structural genes, are regulated. So the repressor is involved in regulating these structural genes. So the repressor represses expression. Okay, the repressor represses expression of the structural genes. And then we have the inducer. The inducer is lactose. So lactose induces the operon only when lactose is present with the structural genes LAC-Z, LAC-Y, and LAC-A be turned on. Okay? If lactose is not present, if the inducer lactose is not present, then there's no need to turn on these genes. Okay, one more thing before I move on. This LAC-I gene, that's the repressor gene, is not in direct contact. It's not adjacent, I should say, to the LAC operon. It's actually found at a very far distance from the LAC operon. That's why you see these dotted lines here. It's indicating that the repressor gene is not it's not a component, I should say. It's not a structural component of the LAC operon, but it is involved in regulating the LAC operon. Okay, so look at that. Okay, so let's see what happens when the inducer is present, and then we'll look at what happens when the inducer is absent. Okay, all right. Well, we'll start with the inducer being absent first, sorry. So in the absence of the inducer, which is lactose, in the absence of the inducer, the repressor protein here, repressor protein, which is produced, encoded, I should say, by the repressor gene, this repressor protein will bind to the operator site. And if once the operator is bound by this repressor protein, it prevents transcription of the LAC-Z, Y, and A genes. How does it do that? It actually prevents the RNA polymerase from binding to the promoter site. See, the RNA polymerase is very large. It's a huge enzyme. And when it binds, it actually needs to overlap both the promoter and the operator site. So it sits on both the promoter and operator site. So when you have this repressor protein bound to the operator site, it prevents RNA polymerase from binding to the promoter. Okay, so if the RNA polymerase is not able to bind, then these three structural genes, the LAC-Z, LAC-Y, and LAC-A, will not be able to bind. Okay, and so it will not be transcribed. LAC-Z, Y, and A will not be transcribed, and hence the operon in essence is turned off. So the LAC repressor is a negative regulatory element. It represses the expression of the structural genes, right? So that's how the LAC operon is repressed. The LAC operon can also be induced. It is induced when lactose is present. So lactose is the inducer of the operon. Lactose or IPTG. IPTG is another substrate for beta-galactosidase, but our focus would be on lactose. So when lactose is present, it acts as an inducer of the operon. It causes the operon to be turned on. So how does it do that? When lactose is present, that's the inducer, it binds to the repressor protein. Once it's bound to the repressor protein then changes shape. The shape of the protein changes. That ability to change is known as allostericism. So the repressor protein is said to be allosteric. It has the ability to change shape depending on what it's bound to. So here the inducer binds to the repressor protein. Once it's bound, the shape of that repressor protein changes such that it is now unable to bind the operator site. Because the changed repressor is unable to bind the operator site, it then makes the RNA polymerase able to bind to the promoter of the operon. RNA polymerase, as I mentioned before, is a large protein, and it binds and overlaps onto the operator site. And once the RNA polymerase binds, it allows for transcription of the three structural genes that are LAC-Z, LAC-Y, and LAC-A. Okay. And note here that the mRNA that is produced is polycistronic. So all three genes are transcribed at the same time. So they're transcribed at the same time, producing one mRNA that has three genes on it. So it's polycistronic. And then after translation, these genes, the components or the proteins are separated from each other into the three individual proteins. Okay, so be sure you understand the difference between repression, how the lac operon is repressed, and how it's induced. So the lac operon is repressed when there is no lactose present, and it's repressed when no lactose is present. The repressor will bind to the operator site and in essence block, and thus, I should say, thus block the promoter, block the RNA polymerase, I'm sorry, from binding to the promoter. That's when lactose is absent. When lactose is present, it acts as an inducer of the operon. When lactose is present, it binds to the repressor protein, changes the shape of that repressor protein, and prevents the repressor from binding to the operator. That frees up the region, the promoter operator region, and allows RNA polymerase to bind and initiate transcription of the structural genes. Okay? All right. So, any questions? Okay, so I'll move on. If I'm not getting any questions, I'll move on. No one? Okay. Okay, so I mentioned previously, in one of the ways that the Jacob and Monod, who are the ones who discovered the lac operon or how the lac operon is regulated, one of the ways they did that was to create mutations and then look at the effects of these mutations. So what we'll look at now are some of these mutations that were created, and these mutations helped in identifying the functions of the various components of the lac operon. So here we have a mutation known as the LAC-I minus mutants. These mutants have a mutant repressor that cannot bind the operator. Okay, so here we have a repressor here that cannot bind the operator. This is your normal repressor. This normal repressor is able to bind the operator site and prevent repress expression of the LAC Z, Y, and A, the structural genes. However, a LAC-I minus mutant repressor is unable to bind the operator site. A repressor that is unable to bind the operator site would allow for continuous expression of the LAC Z, Y, and A genes. So these genes will be expressed in the absence or presence of the inducer lactose. Regardless of whether or not lactose is present, these three genes would be expressed. This type of expression is referred to as constitutive expression, meaning that it is always on. The genes, the LAC Z, Y, and A genes are constitutively expressed. They're always expressed. You could think of constantly expressed in the presence of this LAC-I minus mutation. Okay, LAC-I minus mutations allow for constitutive expression of the structural genes. Here's another type of constitutive mutant known as the LAC-OC mutant. This is a mutation that occurs on the actual operator site. So there's a mutation in the operator. So here, these are OC mutants. And OC mutants, these are unable, they prevent, I should say, the repressor is unable to bind at the OC site, okay? This mutant site. So there's a change here in the nucleotide sequence, and it prevents the repressor from binding, okay? If the repressor is unable to bind, it will again allow for constitutive expression of the structural genes. So no repressor binding means then that RNA polymerase is able to bind at any point, regardless of whether or not there is lactose present, and that would allow for constitutive expression of the structural genes Z, Y, and A. Here's another. This one is called the LAC-IMS. This is a super repressor. Super repressors are repressor proteins that they will bind the operator, but they cannot bind the inducer. They will bind the operator, but cannot bind the inducer. Okay, so a repressor that could bind the operator all the time, it will repress. It will cause continuous, I should say, continuous repression of the structural genes. So this super repressor will cause the structural genes to not be expressed, regardless of whether or not there is an inducer present, because it will remain bound. It will remain bound to the operator. Typically, in a normal setting, with a normal repressor around, if an inducer is added, if the lactose is present, the repressor will preferably bind lactose and leave the operator site free. But with a super repressor is unable to bind lactose. So if you give the cell lactose, the super repressor will not bind the lactose, but instead it will bind the operator site. It continuously binds the operator site, and as a result, expression of these structural genes are always turned off. Okay, so that's a super repressor. Okay. Let's move on. One more thing I want to mention, then I have some questions. Let's see if we could be able to delve into those questions. So it says here, Jacob and Monod used partial diploids to carry different alleles of the LAC regulatory elements and structural genes to identify transactin and cis-actin factors. What are transactin and cis-actin factors? So a transactin factor or transactin element is one that can diffuse through the cytoplasm and act on target sites. So an example of a transactin element will be the protein, the repressor protein, because the repressor protein could diffuse through the cytoplasm and get to the DNA site, in this case, the operator site. Or it could diffuse through the cytoplasm and bind to inducers. So these are transactin elements. They don't stay stationary. They can move around, so transactin. Cis-actin elements, however, are stationary. They can only influence the expression of adjacent genes on the DNA molecule. So cis-actin elements really refer to DNA molecule. It refers to portions of the DNA elements. So in the context of the LAC operon, our cis-actin elements would be our promoter, our operator, the structural genes. Those are cis-acting. They can only influence the expression of adjacent genes. So the promoter and operator are able to influence the expression of the adjacent genes, which are the Z, Y, and A. So those are cis-acting elements. The repressor is not stationary. It can diffuse through the cytoplasm, so that's a transactin element. But here's a question I have for you, just to see if you recall what we talked about last week. It says here that an F-prime LAC plasmids were used to generate partial diploids. My question to you is, What is an F prime plasmid? What is an F prime plasmid? Okay, great. All right. What is another name? I'm gonna type it. So let me just recap. The marrow, you guys are almost, well, I say most of you did answer sort of correctly. So a marrow diploid or a partial diploid is created, yes, when an F prime plasmid initiates conjugation and transfers the chromosomal DNA that it contains with it to a recipient cell, and that new recipient cell now has a partial diploid region where it has it's diploid for just one gene or diploid for just a segment of the chromosome. So that's a partial diploid. So in preparation for Wednesday, I would like you guys to look at figures. Let me get that. Look at figures 15.9, 15.10, and 15.11. Study that, read those figures. So it's, let's see. Yes, 15.9, 15.10, and 15.11. Look at those figures, study those, and the text that surrounds it. And then I will start on Wednesday with some questions that relate to those figures. And you should be able to answer them based on if you did your homework. So that's homework. Make sure you understand that section of the chapter, and we will start there on Wednesday. Okay, so it's now 11:20. I will see you on Wednesday. Bye.